Hello everyone, I am Dr. Iram Jahan Nazir Khan, PG Department of Zoology, Sadhguru Gargi Maharaj College, Karad. Today we will be seeing amyloid beta hypothesis. It is one of the hypotheses in the pathophysiology of Alzheimer disease. So, amyloid beta hypothesis states that the accumulation of amyloid beta fibrils. Amyloid beta fibrils are nothing but the cleaved parts or the cutted parts of the amyloid precursor protein. So we will start with the amyloid precursor protein. What are they? They are transmembrane protein which penetrates through the neuron membrane. So you should remember that they are present in the neuron members and the membrane, and they are transmembrane protein. Next, what are their use? They are useful for the neuron growth. They are useful for the survival of the neurons, and next they are useful for the post-injury repair of the neurons. Next, what happens in the actual mechanism of this hypothesis? Now you all know that the amyloid precursor protein is a transmembrane protein in the neuron membrane. So due to some reasons, unknown reasons, their enzymatic activity, which is mainly proteolytic activity, takes place, which cleaves this APP molecule in the transmembrane of the neuron cell into small fragments. So it is cutted into small fragments. These fragments are called as beta amyloid fragments. So they get accumulate, beta amyloid fragments get accumulate around the neuron cell and form a senile plaque. What is a senile plaque? Plaque is nothing but the accumulation of microglial cells, astrocytes, and these cutted proteins, fragmented proteins, which are dead in nature around the or near the neuron cells. Microglial cells are nothing but the immune cells which are present in the brain region or the spinal cord region, and astrocytes are also the cells, important cells of neuron system which are present in the brain region. So the accumulation of these all types of cells and plus the amyloid beta fibrils together is called as senile plaque, and this is accumulated near the neuron cell due to the cleavage of the APP molecule due to the proteolytic enzymatic activity. Now next, see here, this is a neuron and this is a neuron. The neuron basically shows a neurotransmitter molecule and a neurotransmitter receptors, and synapses takes place. But because of the accumulation of this plaque, the synaptic is not going to happen properly here. So synaptic dysfunction will take place, and ultimately what will happen? The neuron will die. So this is the case in the case of Alzheimer patients, and which leads to the neuronal death. So accumulation of this aggregated amyloid protein causes a toxic effect. It is a toxic form of protein. Second, it disrupts the cell's calcium ion homeostasis. Calcium ion is very important for various processes in the cell. Next, it induces apoptosis. What is apoptosis? That is programmed cell death. Next, the amyloid beta selectively builds up in the mitochondria of the cells of Alzheimer affected brains. So in the Alzheimer affected patients, the amyloid beta also builds up in the mitochondria, and it inhibits certain enzyme functions of the mitochondria of such patients. And also in such patients, it inhibits the utilization of glucose by neuron. So what will happen? The neurons will starve of glucose and they will die. So now you know that because of this hypothesis, the neuron synaptic action is despaired or it is affected, hampered, and plus the neurons die, and which will lead to the dementia and ultimately to the Alzheimer disease. I hope you all understood this one of the hypotheses of the Alzheimer diseases. So thank you.